Hello guys, let's start our lecture and we will talk about the one function, human function, about one subject, but today we have a lot of points. That's why I think that it will be useful for you to get it during the lecture. If you have any questions, please, you have the chat, you can use it. Okay, and later, of course, I will give you the opportunity to watch this lecture in our YouTube channel. Okay, so let me share my screen. So first of all, as you can see right here, so that our, let me just, okay, so the human function. Our learning outcomes for today is, as a result of this lecture, and I hope after that that you will have the ability to discuss it with your teachers. The next learning outcomes: define homeostasis and explain why this concept is central to the physiology. Define negative feedback, give an example of it, and explain its importance to homeostasis. Define positive feedback and give examples of its beneficial and harmful effects. And define gradient. Describe the variety of gradients in human physiology and identify some forms of matter and energy that flow down gradients. So before we'll start this lecture, I want to tell you that this topic is really important for you because this is the general concept of all physiological processes, because we are maintaining right now the homeostatic condition, and we need to have the dynamic equilibrium. What are they? We talk right now. Okay, let's start. Go on. Before we start to talk about this kind of balance and equilibrium, we need to mention what is physiological variation. I hope that you know it from the previous classes, and let's just remind you. Because for different ages, sexes, for different diet, for different patients who have different weight and physical activity, the values, the physiological values in the normal range could be different. That's why you need to think about it. For example, you can see that the last point is over-medication of elderly. It means that for all people, for all patients, they have another metabolic rate, they have another physical weight, another index of the body mass, and that's why you need to consider each dose of medication according to the weight and according to the metabolic rate of the patients. Okay, so the typical example in this textbook, and we will consider the typical physiological values for the, just wait a second, okay? Okay, so guys, let's continue. So you can see that, okay, you can see that in this particular textbook and in all physiological textbooks, if it's not mentioned about the age, sex, or the weight, so we consider the typical physiological values for reference man and reference woman. Okay, and you need to think about it. If you have, for example, if you have patient who has a higher BMI so body mass index. So you need to consider that the physiological variety could be different with this patient. So the next one, okay, let's talk about the homeostasis. I will show you the definition. The homeostasis, this is the body's ability to detect change, activate mechanisms that oppose it, and thereby maintain relatively stable internal conditions. Relatively stable, it means that we have some ranges from one point to another point, and sometimes we have this certain value, and our body will compare this value with these kind of normal ranges of us. Okay, so in the loss of homeostatic control causes illness or death. I want to show it to you. Okay, let's just open. Let's open the. Okay, so look at this, guys. So according to the homeostasis, it means that every time, every moment in your life, you need to maintain the homeostasis. Don't mix it with hemostasis, okay? The hemostasis is just, okay, this is a process to stop the bleeding. It's hemo or high hemo, the same pronunciation, hemo. It means you need to stop the bleeding. Okay, stop the hemorrhage. We need to talk about the homeostasis. Okay, and we have some two types of mechanisms that can help us with it. Okay, we have the feedback loops. Okay, the negative feedback loop, and the positive feedback. Okay. Okay. So the negative and positive feedback, but each feedback, they have differences, of course, and they have particular situation where can we find them. Okay, and but they have certain steps, okay, in feedback loops. Okay, let's consider them. If we talk about the changes in our body, for example, you have the temperature, yes, and you need to maintain the certain temperature. How can you detect this temperature? This is the first step. Okay, the first step of our feedback chain, the feedback loop, will be stimulus. Our stimulus will be the temperature that we are considering. Then this stimulus should be detected by some organ, tissue, cell, and we call it receptor. We have some receptor or sensor. Yes, it will be detected by certain receptors. So then you need to send this signal to one place, okay, because we need to control it. By which organ we need to control it? We need to send it to the control center. This is the third point of our negative and positive feedback. This is the control center. Okay, the control center, in most common cases, it's CNS, central nervous system. It could be the brain or spinal cord. Okay, the control center will do this, the one thing. Okay, the function is comparison. The control center has the certain value of this stimulus. For example, for temperature, in our control center of hypothalamus, in our brain, we have the certain value that we call set point. Okay, set point. So the set point is just the level of this value. the normal range of this value. And control center should compare the stimulus with this set point. So it compares if it's higher or lower than the normal range. So the control center then send the signal to the last part of your negative feedback or positive feedback loop. This is effector. Send the signal to effector. And in most cases, it's the muscles or glands. Okay, so this is just the standard scheme for mechanism. How can you regulate the changes in your body? So we have the stimulus, we have the receptor that detect the stimulus, then we have the some pathways to send the signal to the control center. Then we have the set point, and we compare it to the set point, and then we send the signal to effector. In case of these differences between the normal range and the stimulus. In case of temperature, for example, the temperature is 38, for example, 38. Okay. The 38, it means higher than the set point, because set point is 37 degrees Celsius. Okay, the receptor in this particular case is our thermoreceptors. We have the thermoreceptors, which are peripheral in our skin and the central in our internal organs. And it will send the signal to the control center. In our particular case, it's the hypothalamus for temperature. It will compare the 38 degrees Celsius with the set point of 37 degrees of Celsius. So it means that it's different, and we need to do something, and we need to just take this signal and convert it to the action, okay? And we send the signal to the effectors. In our particular case, it will be the muscles and glands. So we need to decrease the temperature. And we have several mechanisms to decrease the temperature. In this particular case, as you can see, the muscles and the glands. The muscles within the vessels, they will be dilated. Okay, and we will have the dilation of the vessels in our skin, in dermis layer. And that's how can we lose the temperature. That's why when you have the higher temperature, so your temperature has been increased, you can see that your skin are a little bit reddish, because within the vessels we know the blood, yes, and the blood is red color, and when it's dilated, the vessel is dilated, it goes closer to the surface of the skin. That's why we can see reddish color of our skin. And the glands, in our particular case, it's sweat glands. Sweat glands release will eject the sweat, and the sweat, you know, it's fluid, it's water, and that it will be evaporated from the surface of the skin, and that's how can we decrease our temperature. That's the mechanism. And this mechanism, to negate the effects of the stimulus, okay, this is the stimulus, and we need to negate it, so we need to remove the consequences of the stimulus. That's we call the negative feedback loop. Negative feedback loop. So it means that if we consider it as a graph, I hope that you can see my desk, yes, whiteboard. Please, guys, type me if you can see. So it means that this negative feedback. Okay, just let me open the chat. Okay, thank you. So it means that the negative feedback, if we consider the effect as the graph, okay, this is the time, the x axis, and the y-axis is our just severity, okay, of consequences. So first of all, the stimulus was right here, okay, and due to the time of action of the negative feedback, it will be decreased. So that's the action of negative feedback loop. Negative feedback loop. Okay, National Football League. So the you can see that the it negates the consequences. Don't forget about it, okay? It negates the consequences. Okay, so what about the positive feedback? Actually, the positive feedback is not so positive as you could expect. So the positive feedback, as you can see right here, okay, this is the negative. Let's talk about the positive feedback. The positive feedback is the second mechanism that regulates our homeostasis, maintain our homeostasis. But in this particular case, the positive, it means it will add more and more actions after one stimulus. So it's self-amplification cycle. I will show it to you. So, and in your textbook, okay, let's consider it right here. In your textbook, it's mentioned the uterine contractions during the childbirth. Okay, let's consider it, and then I will give you another examples. Okay, of it. So the positive feedback. The positive feedback, it has the same, the stimulus, yes. So let's consider the let's consider the uterus, the pregnant uterus. It's just the process of childbirth. It's already occurred. Okay, it means that the head of the baby, it's right here, the head of the baby, it pushes toward the cervix. This is the cervix, the narrow space. Okay, the narrow space between the vagina and between the uterus, we have the cervix. And within the cervix, this is the stimulus, the push. The push, it's just the stimulus. And you can see that then this stimulus will be sensed by the stretch receptors. And again, receptors, yes, the second step of our feedback chain. Okay, so the stretch receptors, then they detect that the head of the baby just compress this part of the uterus, and then it sends the signal to the control center. Okay, in our particular case, the control center, it's again the hypothalamus. Okay, the hypothalamus. Why to the hypothalamus? Because the uterus is from smooth muscle tissue, and it has the sympathetic innervation, and the center of this innervation of peripheral nervous system is located within the hypothalamus. Okay, so the hypothalamus, it has the connection with the endocrine gland. Which endocrine gland? This is the pituitary gland. I think that you have heard about it. So the pituitary gland located in the okay, and look at this. This is the, I will try to draw the hypothalamus. It's like a mid-sagittal section of the brain. Okay, this is the brain. I'm not an artist, sorry. So, and then right here we have the infundibulum. Right here we have the anterior and posterior pituitary. This is the pituitary. Hypothalamus has the connection with the pituitary gland, with posterior and anterior part. And with posterior part, it has, okay, this is the posterior part. It has the direct connection by the nervous tissue. And through these nerve axons, through these nerve fibers, it will release by the action of these stretch receptors. They will send the signal, and it will release the hormone, okay, the hormone, and this we call oxytocin. Oxytocin. Okay, and the oxytocin has several effects, and one of these effects, it simulates the smooth muscle contraction. And so you can see that in this particular case, in our positive feedback loop, you have the effector as a gland. Okay, and then it has the effector as muscle. Muscle of what? Muscle of the uterus. So the muscle of the uterus, by the action of oxytocin, it will be contracted more and more. And what is the consequence of it? The head of the baby will be moved toward to the cervix again and again. And we have again the cycle. From this point, the head of the baby moves closer to the cervix, and it pushes again the cervix, and then the stretch receptors will send the signal to the hypothalamus. Hypothalamus release with the help of the posterior pituitary oxytocin. Then it will again release the oxytocin, will contract the uterine more and more. So you can see that it's self-amplification cycle. It means that the force of these actions, the force of these consequences will be stronger and stronger. And if you consider again so if you consider look at this. We have considered the negative feedback, yes, negative feedback right here. In our particular case of the positive feedback, loop, it will be another one, okay? If we consider, okay, so the time and severity of the consequences, so you can see that it will be, the stimulus will be right here, and this is the stimulus or severity of the symptoms and consequences. This is the time, horizontal line, horizontal axis, and then it will, okay, the self-amplification cycle. And compare it. That's why we call it positive. Okay, positive, it means it will add more and more consequences to the stimulus. Actually, it doesn't need even the repeat action of the stimulus. It's just one stimulus and then a lot of actions after that. Okay? So it means that till which point, in this particular case of the positive feedback, look at this, till which moment we have the positive feedback and we have the uterine contractions. Before the moment the child's head will go out, that's the point where we will finish it. Okay, so the positive feedback will be till moment when the there is no stimulation, will be no stimulation, it means the stimulus will be changed totally, or you will just achieve some goal. At this point, it's just the child will born. Okay. So, and the next, I hope that you got the difference between the positive feedback and negative feedback. And I want to give you the one example, and you will write me is it positive feedback or negative feedback. Okay. So let's consider it's right here. So think about the lactation. I think that you know that it's breastfeeding. Okay, breastfeeding. When the baby has a suckling out of the mother breast, okay, and the baby will okay, suck the milk. Then we have the stretch receptors, mechanoreceptors of the nipple, and they will send the signal. They will send the signal to the hypothalamus. Okay, to the hypothalamus. Hypothalamus will send the signal to the posterior pituitary gland to release the oxytocin. Then the oxytocin. Then oxytocin has the action on smooth muscular tissue, smooth muscles, and within the mammary gland. Actually, mammary gland is a sweat gland, the modified apocrine gland, and it has the I will draw it for you. It has just the duct, okay, and this duct is out of the two types of cells. We have the secretory cells, okay, which secrete the secretory cells, which secrete and synthesize the milk, and we have another one which is the myoepithelial cells. The myoepithelial cells, they act as the smooth muscle tissue, and in this particular case, oxytocin act on these myoepithelial cells to contract. They will contract and eject the milk out of the secretory cells into the duct, into the milk duct, into the mammary duct, yes. And the next, the milk will be ejected from the mammary gland into the oral cavity of the baby. So, what is the feedback chain? Is it negative or positive? Okay, so the positive feedback, yes, it's positive, totally positive. So the positive feedback, because after that, you can see that the baby will just sense the taste of milk, and then he had the brown fat of the cheeks, and the force of the suckling will be increased. So the mechanoreceptors again send the signal to hypothalamus and to pituitary gland. They release the oxytocin and then ejecting the milk till the moment when the milk will be finished. Yes, the in-born breast. Okay, so the thank you for your answering. I can see it. Okay, so the stretching initiate negative feedback as well. Yes, of course. Let's consider it. I can see your question. These stretching initiate negative feedback as well for any particular hormone. We need to consider one. This is the negative feedback, yes, and okay. For example, this is the positive, and before it was the negative. Okay, let's consider the negative as our example. Yeah. Right here, we have considered the negative feedback when we need to decrease the temperature. But what if we have another stimulus, or we have another type of mechanism? Let's consider how the negative feedback chain could be the positive and could have the harmful effect, and the same for the positive. Okay, so let's start from the negative. Okay, it's about the temperature. Again, I will draw the scheme. Okay, so you know that the stimulus will be the temperature. Let's consider the condition when you have the fever, when you have increased temperature, and in any particular case, for example, for microbial infection, okay, bacteria. You have the bacterial infection and you have the increased temperature, fever. And for example, it's 39 degrees of Celsius. Okay, so the stimulus, of course, will be detected by peripheral and the central thermoreceptors. Then send it to the hypothalamus. And you know, in the hypothalamus we have the control center, and it will compare the this temperature to the 37 degrees of Celsius. Yes. Okay, but in case of the infection, the microbes, they have the specific chemicals, the active substances that we call pyrogens. Okay, actually, what is the they have the pyrogens. What is the action of these pyrogens? It will change the set point for the hypothalamus, and it will change to the 41 degrees of Celsius. So the hypothalamus will detect the 39 degrees of Celsius, which is actually already increased temperature, yes. And but in this particular case, the pyrogens they have changed the set point. That's why it considers that 39 degrees of Celsius it's decreased temperature, and that's why send the signal to the muscles. Okay, to skeletal muscles and smooth muscle tissue. In the smooth muscle tissue, within the vessel wall, it will be the lumen of the vessel will be decreased. We have the vasoconstriction. Okay, so and how can we detect it? Our skin will be pale because the vessel, they will decrease the lumen. They will go deeper from away from the surface of the skin, deeper into the dermis to keep the temperature. That's why we need to constrict it. Okay, in case of the next type of the muscles, as skeletal muscle, the skeletal muscles they will detect the changed temperature, and the pyrogens have changed the set point, and that's why it will be considered as the low temperature. We need to increase it. So the skeletal muscles will receive the signal to contract, and you will have the shivering. Because during the contraction, you will consume the energy, the ATP, and due to this process, you will produce the heat. That's why sometimes, guys, when you have the high temperature, you feel cold, and you feel will have the shivering, and you will have the pale skin due to this process. Of course, there are a lot of reactions, but this one is just changed negative feedback out of the hypothalamus, okay, out of our control center, because the pyrogens act on the hypothalamus. This how the negative feedback could be could have the harmful effect. Let's consider when the same receptors, for example, you asked me about the stretch receptors, will send the signal to have the negative feedback. Okay. Okay. So the stretch receptors, actually, we have the stretch receptors in our smooth muscle tissue, in our skeletal muscle tissue, in our cardiac muscle tissue. Everywhere we have the stretch receptors. What is the receptors, actually? Receptors are the, receptors is just the part of the nervous tissue, yes? Just the nervous tissue, the sensitive fibers of the nervous tissue. We have the motor and the sensory fibers. Okay, this is the sensory fibers that send the signal to the brain or to the spinal cord. Okay, so the stretch receptors within the vessel wall, for example, within the vessel wall, this is the vessel wall. We have three coverings, three membranes, or three walls, as we call tunics. Okay, and the middle one is muscle tissue. Okay, these are muscle tissue right here. And within the muscle tissue, we have the stretch receptors as I drew right here. Okay, so then why we need it? To detect any changes in the blood volume and any changes in the lumen, changing in the lumen of the vessel. So the stretch receptors from the vessel wall could be sent again to the brain, but in this particular case, we will have the brainstem. And we have right there the medulla oblongata, or medulla oblongata. So we have the medulla, and within the medulla we have the vasomotor center. And vasomotor center will maintain the activity of the vasomotion of the vessel wall. If we have increased blood pressure, so we need to decrease it, and the vasomotor center will help us with it. If we have decreased blood pressure, so we need to increase it with the help of vasomotor center, and it will send the signal to the arterioles, to the smallest arteries in our peripheral regions of our body, and it will increase, for example, if it's just drop of the blood pressure. So you can see that it will send the signal to the to the vasomotor center, and it will increase the peripheral resistance. The total peripheral resistance will be increased, and then it will increase the blood pressure. That's the one mechanism of it. But in this particular case, we call these receptors as baroreceptors. Baroreceptors. Why baro? Because they will detect the force of hydrostatic pressure on the vessel wall. Okay? That's how the same receptors from the same tissue could be detected and involved in the negative feedback mechanism. Okay, let's consider the next example, the harmful effect of the negative feedback. For this moment, I need to open it. Okay, just let me show to you the. Okay, so as you can see that the harmful positive feedback loop. The fever, again, the fever. I told you about the pyrogens action, yes? And you can see that it's involved, the fever involved in many mechanisms, many responses, and the metabolic rate. Metabolic rate increases, body produces heat even faster due to the metabolic rate changing. And then the body temperature continues to rise and then further increases the metabolic rate. This is the harmful positive feedback loop. And you know that the body temperature could be increased by the and it for our metabolism of the proteins especially, it's really harmful because it could be denaturation, yes, denaturation of proteins. The cycle continues to reinforce itself, becomes fatal at this point of the Fahrenheit, degrees of Fahrenheit. Okay, so this is the harmful positive feedback loop. Let's consider the next one. If you have any questions, you can ask me in the chat. The next one, what we should consider, the gradient. Okay, but before it, I want to just talk to you about the mechanisms of the transport. The mechanisms of the transport, the first mechanism is diffusion. On this particular slide, you can see the facilitated diffusion, but we have the simple diffusion too. What is the diffusion? I think you know it. Okay, just let me add the slide to this. Okay, and okay, the simple diffusion. The simple diffusion, it means that okay, we have just maybe semi-permeable membrane. Maybe we have just two portions of gases. Okay, for all we have the fluid and the gas, and we have just the certain amount of the molecules in one side and in another side. So the diffusion, it means that it should balance the concentration between two media, between two sides, yes, and it will go to this side before it will be equal, before the moment when it becomes equal. So without any sources of energy, it doesn't require any energy. The second type of the diffusion, it's just this facilitated diffusion. It means that for this movement of the particles, of the substances, the chemicals, from, for example, in this particular case, from ECF, extracellular fluid, to the ICF, intracellular fluid, from outside to inside the cell. So we need to have some special proteins, okay, the carrier. Okay, you can see that the membrane protein and carrier helps us to provide the movement from outside the cell to inside the cell, but without any sources of energy. It doesn't require any energy. That is facilitated diffusion. And actually we have this for our glucose, okay, to move the glucose, to have the glucose transport from outside to inside the cell and vice versa. So we have this the sodium glucose transporter. Okay, the next one. The next is filtration. I hope that you know it. Yes, filtration occurs in capillaries. We have look at this. This is the capillary wall, and we have the endothelial cells. Okay, and between endothelial cells, we have just look at this one. Okay, the pores, yes, between them. The spaces, the gaps between the endothelial cells in capillary, they are for filtration. And you know that we have the capillary exchange. How can we have the gas exchange according to our respiration? Yes. So we need to filtrate something. For example, in this particular case, you can see the solutes. They will go through the capillary wall with the water, and you will see that it's due to the hydrostatic pressure. Okay, I will draw it. So the hydrostatic pressure, in this particular case, it's pressure of the blood. The blood pressure compress the fluid and move it toward the wall of the capillaries. And in the capillaries, okay, in the capillaries, we have the pores, we have the gaps, and through these gaps, just some substances will go out with the water, and we call it just the filtration. The huge particles, as the red blood cells and the proteins, they will stay within the vessel. They will not go outside. They will stay within the vessel. Okay, this is the filtration. And the next one, the next membrane transport is osmosis. Osmosis is just the movement of the water. Okay, the movement of the water. Water flows through the cell membranes because the cell membrane is semi-permeable membrane, and we have particular amount of the solute, the particles of the solute, and we have the water. And you know that the when you have the more concentrated area, the water will follow for the more concentrations to dilute it. Yes, to dilute it. So it means that in 30 minutes later, in the same in the same vessel, we have the equilibrium. The equilibrium of osmotic pressure, and we have the decreased hydrostatic pressure. We need to just have more water if it's more concentration according to osmolarity. Okay, so it means that if we have the high concentration, the water will follow, and to maintain the balance from the side where it's more concentrated to the side where it's less too. And you can see that the osmotic gradient is really, really important. This is just the ratio between the concentrations, between the amount of particles of one substance to the amount of particles in to another side, and you can see that down the gradient, it means that the down the gradient, it means, look at this, we don't need to have any we don't need to have any sources of energy for it. We will move down the gradient, and from the to have equal concentration between two media. And if you are talking about the up gradient, it's like climbing the walls, climbing the mountains. You need to have some source of energy. And actually, in our body, we have a lot of the matter that flows down the gradient. For example, look at this. Okay, look at the blood. It flows from the area with a high concentration to the area to the low concentration. To the if it's about the pressure, so from the high pressure to the low pressure. If you talk about, for example, the digestion of any substances, lipids. Okay, the lipids from concentration of the from high concentration of lipids to the area of low concentration. It's how can we move them through the cell. Okay, so the next one, you can see that even the glucose. Okay, the glucose. But for the glucose, we need to have some co-transporters. Yeah, so we need to have some membrane proteins to provide this movement, but without any energy. It's down the gradient from the high concentration to the low concentration. Okay, and even the heat, the temperature, how can you lose the temperature? From, you know that if we consider, okay, this is the body, the outside temperature in normal conditions, it's lower than your body, yes, and according to the heat, of course, it will be another one. Okay, and you will evaporate the heat. Okay, due to the several processes as conduction, convection, evaporation, and you will lose the temperature of your body. Okay, that's why during the winter we need to cover to keep the temperature, to decrease the loss of the temperature. Okay, so and it will be due to the gradient, down the gradient. Okay, and that is all that I needed to discuss with you. Just wait. Okay. So the I can see that you wrote something for me. Is that how then glucose decrease or increase, the insulin, the glucagon regain back? Okay, it's about the hormones. Little bit another one. Okay, because the glucose. Okay. The glucose transport is the same one, yes. We will reabsorb it in our renal tubules. We will move. We can store it by our liver, by muscles. We can convert it as a fuel into the ATP, and we can make the glucose from another types of fuels as the fatty acids and proteins even. But in this particular case, we are talking about the neuroendocrine reflex. Okay, so let's consider it. Okay, little bit. It's actually it's not the topical for you right now, but I will. Okay, we'll just show it. Okay, so you can see, I think, the whiteboard. Yes. So we will try to draw. Actually, if we consider the control, control for our for our body to maintain the homeostasis, yes, we have two different systems. They are connected to each other tightly. I will show it to you right now. We have the nervous tissue, the nervous system, okay, and we have the endocrine system. Okay, and that's how can we control it. We have a lot of similarities between them and the differences. But the first difference between them, they use the hormone. The hormone is the tool for endocrine system. For nervous is just the nerve signal. Yes. And nervous tissue receptors. Okay, so, and if you talk about the glucose, so we have the pancreas. It's exocrine and endocrine gland. It releases the hormones as glucagon and insulin. Okay, and they are antagonistic to each other. So insulin helps us to store the glucose and in the liver and convert it into the glycogen, and stores in the muscles because the muscles need it for the contraction. And glucagon will do the vice versa, opposite action, antagonistic action. It will remove the glucose and convert it from the glycogen and convert it into the glucose and release it into the bloodstream. Okay, and we call this kind of we have two different relations, two different tools to control. We have the nervous, okay, neuroendocrine reflex. Neuroendocrine reflex. According to the hormone, when we have just hormones without any nervous tissue, so we call it the negative inhibitory feedback. Okay. For example, in neuroendocrine reflex, the glucose level will be detected by receptors. What are the receptors? Receptors are the nervous tissue. Okay, this is the nervous tissue. It means they have the neuroendocrine reflex. They involve the nervous tissue. It's not just the hormones. If we talk about the hormone, we can take the thyroid gland. We have the hypothalamus, and it will release one hormone that acts on the pituitary gland. Okay, I will draw it for sorry. The hormone from hypothalamus, then it goes to the pituitary gland, to the anterior pituitary gland, and to the thyroid gland. And if we release a lot of thyroid hormone by the thyroid gland, so this actually hormone acts on the pituitary and acts on the hypothalamus to inhibit it. That's the negative inhibitory feedback. That's how can we regulate the level of the hormones. But it doesn't involve any nervous tissue. It involves just the hormones. That's the negative inhibitory feedback. The one particular mechanism, how can you control the releasing of the hormones and the level of the chemicals in our bloodstream, in our body, extracellular, intracellular fluid. About the nervous, we have the neuroendocrine reflex. According to the thirst, for example, if we have any, according to the positive feedback, we need some neuroendocrine reflex because we have the stretch receptors, the mechanoreceptors, which are the nervous system, yes? And then we will release the oxytocin, which is hormone. So the neuroendocrine reflex just occurs there. So I hope that I just explained you something that helps you to sort out what is it, what does it mean. Okay, so the for this moment, okay, so I hope that actually, guys, thank you for this lecture. I hope this will be the main concept for you. We will repeat it and repeat it till the end of your clinical practice. You will think about the receptors, stimulus, about the control center, and how can you decrease the consequences of the stimulus. Sometimes, in the case of positive feedback, the stimulus is already not important. You need to fight with consequences just, okay? You need to stop the positive feedback loop. Okay, hope it was useful for you. I will convert it and attach it to our playlist. Thank you. You can leave this conference, and bye. Good luck.